and Ness for inviting us. So we are going to talk a little bit about the Landscape Ecology programme. I'm going to give a broad overview, and then Laura is going to get stuck into the science, and that reflects our respective disciplines. My background is landscape management and horticulture. I'm the director of WACO, so I'm very much a generalist here, and Laura is our expert mycologist. So what I wanted to do is just use some images of WACO to kind of exemplify the WACO itself, and then the sort of the broader purpose of the programme. And we'll start with this nice overview. So you, with drones, you can kind of give a sense of kind of vast, endless spaces. And this is Southeast England, so it kind of challenges perhaps some preconceptions of our part of the country, and perhaps challenges some perceptions about Kew as well. So Kew is often known, I guess, for one of two things. It's a famous garden in southwest London, which attracts nearly 2 million visitors a year, or our really quite global research programme. You know, we're a kind of biodiversity research institute. But there are other aspects to Kew, and this is our site in Sussex, which is a sort of more semi-natural landscape. It's called Wakehurst. It's 535 acres. It's a really nice kind of mosaic landscape with lots of different land use types. And really intriguingly, we're now applying science to start to understand this landscape better. And indeed, the sort of the broad headline for the Landscape Ecology programme is that we want to kind of research, share and engage people in the value of UK biodiversity. And we try to do that through kind of 4 kind of distinct research pillars, which I'll get to in a moment. But just a little bit more about the landscape itself. So we have some designed elements. So this is an American prairie, which we created. So we're really into grasslands, and we take quite a global view of grasslands. We have our own acid species-rich grasslands, which are very representative of the high Wealds landscape that we're on. So a relatively low amount of species because our soil is quite fertile, but kind of just an absolute kind of sea of nectar in terms of the flowers that grow out through the grasses in high summer, and that supports a fantastic array of invertebrates. But we also recreate grasslands using other floras. So this is an American prairie in its very, very early stages. It will eventually become dominated by grasses and long-term forbs. And our next project is a Silk Road steppe using the grassland floras of Kyrgyzstan, which will be an interesting diversion for us. These are the acid kind of species-rich grasslands that represent the kind of UK habitats that we have. And we've got you know, acres and acres and acres of these grasslands, some of which are really quite historic. In fact, one we know has been around for over 100 years. We also have woodlands of different types. So we've got woodlands of the world. We've got a really broad temperate flora at Wakehurst, so we can grow trees from Chile, from Tasmania, from New Zealand, but also from sort of East Asia, so from China and Japan. And this allows us to sort of ask some interesting questions about ecosystem function and ecosystem services of UK and non-UK tree floras, because one of the really alluring things about Wakehurst as a research site is that we have these kind of compartments, these research plots side by side. So we've got an English coppice woodland sat beside a kind of a Chilean woodland of these Nothofagus. So here's that coppice woodland. So this has been around since the 14th century, and here's the temperate woodland of Nothofagus species, which has been around since the kind of 1980s. But yes, big enough plots to kind of generate meaningful research data. So some interesting questions about UK and non-UK flora. We're very broad-minded in terms of the roles that these different species will have, especially as some of our kind of key UK tree species, especially Fagus sylvatica, appears to no longer be fit within our specific site. So here's that coppice woodland, and this is actually one of our kind of main research plots. And then more broadly, we have nature reserves. So this is where we focus solely on UK flora, and we work very much to habitat classification, and we are in pursuit of kind of good condition here. These tend to be well cared for, good condition, nice representative kind of habitats. So, you know, ancient woodlands and grasslands. And we've got about 150 acres of sort of kind of purely UK habitat. So the landscape ecology program was, I guess, initially sparked by Professor Cathy Willis, who was our director of science at RBGQ. She was also on the government's natural capital committee, and she was very keen that the Waker site was used more to sort of generate, I guess, interesting insights into the kind of ecosystem services and societal benefits that UK biodiversity could provide. And really for a set of reasons. So one, the site was diverse, this kind of set of compartments of different habitat types. The site was in good condition. The site was well used by people. So there's the opportunity to kind of research the cultural services of a UK kind of biodiverse landscape. And we had complete access and complete control of the site. So from a scientific point of view, there was never an issue with getting on the site and getting into sort of specific corners. And the way kind of Cathy helped us to frame it was to kind of think about multiple services and benefits at any given moment, and never to become too preoccupied. The obvious headline is is carbon, but obviously if you become too preoccupied with carbon, you perhaps miss the kind of the multifaceted benefits of nature. So we framed it in these kind of four ways. So to think about climate adaptation and mitigation, primarily how biodiversity can sequester carbon and also adapt to a changing climate, nature connectedness, and really how nature makes people feel, the influence of nature on well-being and mood, hydrology, the idea of biodiverse habitats kind of adapting and mitigating to flood and drought, and then finally kind of pollination and this kind of question of sort of pollinators in the landscape and the specific sort of services that they provide and the way that the landscape can support them. And then we sort of frame it through these success measures as well. So, we are a high impact research institute, so we do need to produce meaningful and innovative research, but we have a role within DEFRA. We're an arm's length body within DEFRA. So we also want to use valuable insights from government managed land to kind of shape those kind of landscape policies, linked to the Environment Act, which are going to become more and more influential. We want to connect people to nature and kind of always think about the multiple audiences that use the site, be they school groups, visitors, members. And then sort of finally, the sort of sharing best practice. There's a very practical nature to what we do. We can form a good affinity with fellow land managers and talk to them as peers. And kind of perhaps provide evidence for changing land use in a positive way that could be valuable. So just very quickly, there's a fundamental commitment to measure the site in detail and to really use the fact that we are here on this site for the long term to go into depth and have this mentality of us being a sort of a living lab, a super site, analogous to how sort of Whiteton Woods is used by University of Oxford, that we always think about these multiple audiences. So for instance, our nature connectedness research, we give our members heart rate monitors and we ask them to step out in different parts of the landscape and record how they feel objectively and subjectively. With our school groups, we had over 1,200 school children taking part in an experiment last year where we basically prescribed them a different habitat to go and spend time in, a wetland, a woodland or a meadow. And we then use kind of pre and post survey questionnaires. And we actually work with Royal Holloway, University of London with their psychology team to kind of develop a robust methodology for seeing how these habitats made them feel. And meadows emerged as a sort of strong winner in terms of being beneficial for well-being and mood. And in terms of pollination research, and actually it's invertebrates more broadly, and it's not just about pollination, we're looking at questions around how trees can be really kind of positive invertebrate habitats, and looking particularly at the role of kind of tree nectar and even tree pollen, and even wind pollinated trees do have a significant kind of provisioning benefit for invertebrates. We're also interested in things like natural pest control. And then sort of onto this kind of, I guess at the moment, our most significant research strand in terms of sort of funding, which is around carbon. And what we're going to talk about in a little bit of detail today is, I guess, kind of fueled by this project. So this was money that we got through Treasury, which is one of the great advantages of being an arm's length body. It allows us to access research funding and kind of government funding. We got together with Natural England, with Environment Agency and with Forestry Commission to really look at how we can resolve some of the tensions between the net zero targets on one side and the kind of biodiversity abundance targets on the other. and to not be too preoccupied with sort of short-term carbon uptake, but really ask the question, you know, where is the carbon in our current biodiversity, and do we have really well measured baselines for all of our current habitats? And Kew's particular interest was to take a very holistic view of the relationship between biodiversity and carbon, not just above ground, but below ground. And this is where Laura will come in. So just to sort of briefly talk about the approach we take to measuring carbon. So we call it a vertically integrated approach. So this is thinking really about spatial analysis, gas flux, and mycology. So the spatial analysis is just taking advantage of having a well-resourced research team on one site and trying to really go to a sub-millimeter scale in terms of measuring above ground vegetation and understanding where the carbon stocks are, laser scanning specific trees, to really kind of get to the bottom of the sort of the relationship between above ground mass and carbon storage, and testing some of these really kind of ground truthed approaches against well-known metrics such as the Woodland Carbon Code, and ultimately trying to say, well, is there another degree of accuracy we should be applying to these metrics, which are ultimately leading to some very major kind of global financial movements in terms of carbon trading. The gas flux is about, as the name suggests, trying to measure the shorter term movements of greenhouse gases in and out of the landscapes. And we look at 4 different habitat types, 3 different types of woodland, including a conifer plantation and one of those species-rich acid grasslands, which I showed you. And then mycology, this kind of really significant kind of missing piece of the puzzle, something that is consistently overlooked when people talk about carbon storage. And at this moment, if it's okay, I will hand over to Laura to sort of talk about mycology in more detail.
Thank you, Ed. Let's see if I'm able to share. Can you see my screen?
Yeah.
Yes.
Yes.
Well, thank you for inviting us today. I'm Laura Martinez Suz, and I'm a research leader in mycology at Kew. And as Ed mentioned, I'm part of the team that is working on carbon and biodiversity below ground. We are focusing on a group of fungi that are called mycorrhizal fungi. I didn't know if you were familiar or not, so maybe this is a little bit simple for some of you. Please forgive me for those of you who know already, but we are working with a group of fungi that are called mycorrhizal fungi. These fungi live in symbiosis with the roots of 90% of the plants we know, and it's a mutualism where both plants and fungi benefit. So the fungi that are associated to the roots of these plants explore the soil, uptake water and nutrients, and in exchange, the plants transfer carbon, which means that these fungi are directly involved in plant nutrition and growth, but also they are sinks of carbon from the plants that are pumping into the soil. So they have a contribution on carbon storage and sequestration in soil, and I will talk a little bit more about that later. So why do we focus on this? Not all the plants form the same types of mycorrhizas. There are 2 different types. Ectomycorrhizas, and that is when the fungal filaments cover the fine roots, but they don't penetrate the root cells. And then there are endomycorrhizas, that is when the fungal filaments get inside the root cells. There are 3 main types of endomycorrhizas: arbuscular mycorrhizas, ericoid mycorrhizas, and orchid mycorrhizas. And I will talk a little bit more about those. So these different types of mycorrhizas, they have differences in anatomy. Different plants and different fungi form them, but also they have different evolutionary histories and different functions, which makes this important for us to understand ecosystem functioning. For instance, arbuscular mycorrhizas are dominant in meadows, crops, and in some tropical systems, and they penetrate the root cells, and they form these tree-like structures that you can see at the bottom where the hyphae branch, and then is where the exchange between the plants and the fungi takes place. These are very abundant. 80% of the plants form these ones. And these fungi are specialized in the uptake of phosphorus. Ectomycorrhizas are the ones that form the little like sheath around the fine roots, and they are dominant in forest, in temperate and boreal forest. Most of the trees in the UK form, or some of the dominant trees form these associations, and they are specialized in the uptake of nitrogen. Within this type of ectomycorrhizas, there are fungi that are specialized in the uptake of inorganic nitrogen and other ones in the uptake of organic nitrogen. In heathlands, the dominant mycorrhizal type are the ericoid mycorrhizas, and they are named because they associate with plants from the Ericaceae family, like heathers or vaccinum. And in these usually nutrient-poor environments, they give the plants a competitive advantage because they are able to unlock nutrients from organic matter. They penetrate the root cells, they are endomycorrhizas, and they form coils. You can see some of these coils in the photo. And then finally, the orchid mycorrhizas, these are quite particular because they are essential for the orchids to germinate and also in the achlorophylltic stages, which means that they are not only taking carbon, but also nutrients. So this is how important these different mycorrhizal types, and they are present in different abundance in different ecosystems. Sorry. So why do we study mycorrhizas? In the last 10-15 years, there are studies that have shown that fungi, and in particular mycorrhizal fungi, are key drivers of the global carbon terrestrial cycle. This is a study published in 2013 where they saw that 50 to 70% of the carbon in soil and forested islands in Sweden was deriving from roots and roots-associated fungi. So they were highlighting that these fungi are regulators of ecosystem carbon dynamics. Also, there are experiments where they study the influence of carbon dioxide in growth of plants with different mycorrhizal types. They saw that plants that harbor ectomycorrhizas, those ones that cover the fine roots, can keep growing despite nitrogen limitations when carbon dioxide increases, while plants that have endomycorrhizas or arbuscular mycorrhizal fungi, they stop growing. This shows that mycorrhizal type needs to be included in models if we want to understand and predict the impact of climate change in these in plants and the response of plants to climate change. And finally, these were experiments, but also there are meta-analyses that show that this effect of mycorrhizal type on the carbon cycle is large and is independent of the effect of many other factors that are important for soil carbon storage, like net primary production, temperature, precipitation, and soil clay content. And this holds at the global scale and across different biomes. They also found that, for instance, systems where ectomycorrhizas or ericoid mycorrhizas are present store more carbon per unit of nitrogen than systems that have endomycorrhizas. So it is important to take into account mycorrhizal type when we are trying to understand the carbon cycle. Why is it relevant now? We found this report from 2021 from Natural England, and is evidence for carbon storage and sequestration by habitat. And we found that, well, they highlight that soil is a big black box and that there is a need of a methodology that allows to measure carbon across different habitats. So measurements can be integrated in a single model and also can be compared. And this is just an example. This graph shows on the right is net carbon loss, on the left net carbon uptake, and here there are some semi-natural habitats and production systems. And you can see how they vary, the length of the bar charts and also the length of the error bar. So there is a lot of variation across habitats, but also within habitats. And if as an exercise, we would map the type of mycorrhizas and the different proportions of these mycorrhizas within habitats, maybe we could explain these differences and large variation across and within habitats. And this is why the abundance and proportion of mycorrhizal types matter. It is important that we understand the contribution of these different mycorrhizal types to carbon sequestration in these habitats. For many years, we have been working mostly in forests across Europe and the UK, and these dominant forests, oak, beech, spruce, and pine, harbor ectomycorrhizas, the ones that cover the fine roots. And we were able to develop a method that allows us to identify these fungi in the roots, but also identify large-scale environmental predictors of their diversity. So we know that these different mycorrhizal types are important, but also within each mycorrhizal type, the community composition is important. So we were able to find fungi that are indicators of different environmental conditions and thresholds at which these communities change. But the most important thing is that we set up a below ground baseline that can be used to assess future change in these forests. We found that when mostly nitrogen deposition, when it increases, it changes the community composition. Some fungi decrease or even disappear. And when this happens, the mineral nutrition of the trees gets affected. So there is the tipping point at which forests or trees transfer from being nutritionally balanced to being nutritionally imbalanced. And this is because, some of these fungi that have certain functions decrease or even disappear because of nitrogen. So it is important to understand how communities change within mycorrhizal type. And these fungi that are the ones that decrease or even disappear are the ones that are able to pump more carbon into the soil, which means that this can have further consequences in carbon sequestration. Why are we doing this at Wakehurst? As Ed explained, Wakehurst is the great place. It can be used as a living laboratory and as where we can develop methods and where we can study how these habitats store carbon more in depth. So what we did was to adapt the method that we develop in forests to woodlands and meadows at Wakehurst. We have been measuring soil carbon, nutrient and biological data, and I will get into that in my next slides. We have done this across seasons and habitats, and the aim is to integrate all this data, not only the below ground, but also the data at the ground and above ground level into a model so we can understand how these habitats store carbon and how these also relate to biodiversity. We are doing this in a meadow and 4 different broadleaf and conifer woodlands. And we are measuring soil carbon and nutrient data. We take soil cores and we analyze soil chemistry, but we are also collecting soil biological data. And this is assessing direct below ground mycorrhizal type proportions. So we extract the roots from the soil and we assign them to these different mycorrhizal types. We also, in the ones that have endomycorrhizas, the ones that have the fungi inside the root cells, we assess root colonization. And this will tell us the extent of exchange between both hosts and how much biomass is in the roots, fungal biomass. We are also identifying through molecular methods the fungi in the roots. We are quantifying the biomass, the fungal biomass in soil. And we are also, when possible, assessing functional traits. We are trying to also record traits related to nutrient forage or carbon sequestration. And thanks to the work that we have been doing and the method development at Wakehurst, we are now contributing with our research to the soil monitoring efforts that are already taking place as part of the Terrestrial National Capital and Ecosystem Assessment Program. This is the team that is involved in this research, and the aim is to improve and inform assessment of ecosystem condition. This is across England, different habitat types. Forest research is sampling forests, and Natural England are sampling other habitats. And the idea is to improve ecosystem condition related to carbon stocks and nutrient cycling, also select below ground indicators of carbon sequestration or other ecosystem processes, and improve and inform the decision making around land management, conservation and habitat prioritization. But mostly the idea is to generate the baseline for soil and carbon, soil carbon and soil biological data that can be used for long-term monitoring. Thank you. And I'm